just briefly, my lab is generally interested in mutational processes. Okay, so you've perhaps seen pictures like this. You sequence the genome, and then we'd like to figure out where the mutations are, and then understand them. So the subject of today is really a sort of more generalizable mathematical way of thinking about these mutations. Okay? If you are totally new to this field, you might want to look at a review paper that I wrote with other people in my lab. It came out in December of last year. In the early days of genome sequencing, we really talked about point mutations, like one base pair changes and indels, and then copy numbers. But really, there are a whole lot of things that you can find. And so this is still a pretty active area. And there are lots of things that are not possible with short read data. But we're getting better. And we now have long read data to use as benchmarking tools, right? And so we actually can develop short read based tools and then figure out whether they're correct or not. Okay, genomic data analysis, do not believe everything you read. This is really true and becoming truer because people have so much data, they have some favorite conclusions, and when you have a lot of data, it's easy to see what you want to see in the data. So these are some of the papers that have been published. And these are the papers that we wrote saying that the papers on the left are wrong. Okay? And there are lots of stories I can tell you about these. But these are published in what I call vanity journals. Okay? So people tend to like these journals. But really, this paper, for instance, there was a mistake that a graduate student made in coding. Okay? But they had a conclusion that was different from the paper that we had published 1 year prior to that. And so we had to go through their code, and it turns out there was a little one line of code mistake that gave them a very different result. I can go on, but I'll just skip that for now. One of the things that some people in this department work on is mutations, what's sometimes called mosaic mutations. These are somatic mutations. The term is almost synonymous with the mosaic mutation is almost synonymous as somatic mutation, but we say mosaic because it sort of emphasizes the fact that within a population there are many cells with different types of mutations. Okay, and so these are mutations that are harder to detect. So there are a lot of algorithms for detecting mutations in cancer samples. Those are a little bit easier relatively easy, because cells with those mutations tend to have proliferative advantage, right? So you have more and more cells with that mutation. Whereas in other tissues, that's not the case. And so those are harder. So it's a more fun computational problem. So we spent a lot of time trying to find these mutations that are harder to detect and to use single-cell DNA sequencing approaches. And so you probably have heard of a lot of single-cell RNA analysis. So that's pretty common now. It's pretty easy to do. But single-cell DNA is much harder. Okay? So I can give a whole talk about that, but I'll just say that it is a different approach. It is expensive, right? That's the main limitation, right? To sequence one human genome would cost as much as sequencing one cell in that person. So if you want to sequence 1,000 cells from one person, that's like sequencing 1,000 genomes. Anyway, so there are ways to do bulk sequencing. Bulk just means population of cells. And then we can also isolate neurons and then amplify them in sequence. And so we spent a lot of time trying to figure this out. Okay, so the main topic is mutational signature analysis. I'll try to motivate that by giving you some examples. I think most of you know that lung cancer and melanoma have different mutational profiles. Okay? Lung cancer, if you just sequence a patient with lung cancer, you see a lot of C to A substitutions, a lot of C to Ts. If you sequence melanoma samples, you see a lot of C to T, but not much else. Okay? If you sequence gliomas, okay, you actually see something that's similar to melanomas. Okay? A lot of C to T. Okay? But the sources of the mutations for these two cancer types are very different, right? Well, it turns out that if you look at not just a single base, but its neighboring bases, okay? Trinucleotide context of the substitution, then you can actually tell them apart. Okay? And so rather than just looking at one base pair change in its isolation, we're going to look at its context. So we look to one base to the left and one base to the right. And because of you have four on each side and six, it's six because the opposite strands are hard to distinguish. And so if you look at the brain cancer samples and you look at 96 dimensional vector, right? So for C to T, I look at all the neighbors. Okay? So that gives me 16 here. Okay? So if you look at the 16, so still the substitutions, single base substitutions, but its context, then they look very different. Okay? So each mutational mechanism tends to generate mutations in a different context, different trinucleotide context. Okay? And so the idea is that we're going to use the context to figure out what is causing the mutation. You can ask the question, well, why just one base? Why not go further out? In fact, you could do that. You can look at two bases, and you can sometimes tell it apart, but the amount of information goes down as you go further out, and the complexity of the calculation becomes greater. Okay, so how does this work? Well, it is actually pretty complicated because when you sequence a tumor, it's not just one process, right? I mean, I've given you the melanoma case, which is probably the simplest. But in fact, you have multiple processes. They act at different times for different durations, right, on different groups. And so suppose you have three biological processes. Okay? And then you make an observation. Okay? So I sequenced a cancer case, assume that there are these three processes, I make observations. Well, I can't figure out what those three processes are, right? Because I only have one sample and three possible processes. Okay? But if you have a lot of samples, okay? If you have 100 samples, 1,000 samples, you can actually start to tell which are the underlying processes. Okay? So that part is the mutational analysis part. Okay, so how does that work? Okay, if we have enough observations, then we can use some pattern recognition algorithms to discover the signatures. Okay, it's just like listening to a band play. If you listen to it for 1 second, you can't tell which are the instruments. But if you listen to it long enough, okay, you can actually tell which instruments are present. Right? It's the same idea, same algorithms. Okay? So it's called non-negative matrix factorization. I'm sure in your sophomore or freshman class, you've learned matrix factorization. This looks fancier only because of this non-negative part. Okay? It's non-negative because each of these processes should have a positive coefficient, right? So that's the only complication. Okay, now, this was all new in about, 2014 or so, 13 or so. Okay, so here's a paper, the first paper on this topic. What they did was take all the samples that they could find, 500 whole genomes, 6,500 whole exomes, and then did this matrix factorization. They said, what are the underlying building blocks that make up the signatures? And so they came up with 30 or so. Okay, a couple of years ago, this group of people, International Consortium of Cancer, International Consortium of Cancer Genome Consortium, International Cancer Genome Consortium, of which I was part, came up with a bigger catalog, although I wasn't involved in this particular process. And so every time you have more and more data, you have a greater power, statistical power to find these signatures. And so the catalogs keep getting larger and larger. And so there are 85 signatures on this one. Okay? In fact, when you have more data, you can do double substitutions and indels, and now you can do copy number and structural alteration, signatures. Okay? Now, one of the things that you want to do after you get a signature is to figure out what each one corresponds to. And so people have spent a lot of energy trying to figure out what those are. And so DNA repair deficiencies, polymerase errors, etc., they all have specific signatures associated with that. Okay? And so, I mean, you can imagine doing a mouse experiment where you expose the mouse to some mutagen and then see what the mutation profile looks like and then link the two, right? Okay. So that process of trying to look at the spectrum that came from multiple processes and then doing some matrix operation and coming up with signatures is the subject. Okay? And it turns out it's useful for everything. It's one of these things where after you see the paper, you realize, my goodness, this is such an obvious idea. Why didn't I do it, right? Okay, so I'm going to show you two examples of how we've worked on this, improving this. So one is to discuss how to do this in the context of panel sequencing data. So panel sequencing is what is typically done if you go to a hospital right now. So any major academic hospitals in the US, many of the hospitals in Korea, if you have cancer, they might do a panel sequencing. Okay, so you hear a lot about whole genome sequencing. My lab had been doing whole genome sequencing. That was our main thing. I mean, when I started seeing whole genome data, I thought, wow, this is going to be the way of the future. We're going to develop algorithms for this. But it turns out that panel sequencing is what still goes on in the clinic. Okay, because whole genomes are much harder to interpret. Do you have is there a clock here? Oh, I see. I'm sorry. Okay, I see it now. I just want to make sure that I'm not keeping you here too late. Okay, I'll describe some ideas that we had for panel sequencing data because I thought being here in this department, there might be many of you who are interested in not fancy analysis, but what we can actually do in practice, okay, with the things that we develop. Okay? Sigma, we've actually have licensed it to companies like Pfizer is using Sigma now for some of their analysis and so on. So I'll describe why they're doing that. Okay. The work that I'm going to talk about are done by Doa and Hu. Doa is an ex-physicist. She came to MIT for a PhD in experimental particle physics. And she was working at CERN and then decided that she should try something else. She's extremely good with algorithm development. And Hu also has a physics background and is superb. Okay. Of all the signatures that I told you about, there's one signature that's really important for this project. That's signature 3. Why is it 3? Because it just happened to be the third one. There's no reason that there's no other reason that it's signature 3. Single base substitution because we're still talking only about point mutations, right? Now, this signature, I mean, these bars look kind of random, but they're not, okay? This pattern is found in BRCA1 or 2 mutant tumors, okay? So there are many ways that people have shown that this signature is related to BRCA mutations, right? You can actually do CRISPR gene knockout, okay? And so you can, wild type has this, but if you knock out components, okay, of that pathway, like here's BRCA1 and 2, the mutation profile that you see just looks like signature 3. Okay? Now the problem is that this signature is harder to detect. Okay? If it has a peak at some of the bars, but empty in other bars, it's a little bit easier to detect. But because it's relatively flat, it's harder to detect. Now, why are we interested in detecting signature 3? Because if you have signature 3, you should be considered for getting what's called PARP inhibitors. Okay? I'm not going to tell you much about how this actually works, but this is a relatively new class of drugs. There are tons of papers, 134 trials right now, but this was a while ago. There are a lot more. There are major companies all developing PARP inhibitors. Okay? And so if you can figure out which cases have BRCA1 or 2 mutation in right now breast cancer, you are eligible for a PARP inhibitor. But what we thought was, it's actually not just BRCA1 and 2 in breast cancers, but lots of other genes that might give the same phenotype. Okay? So in fact, you can actually sequence BRCA and find mutations, but there are other genes in the same homologous recombination recombination pathway that can lead to HRD. Okay? So for instance, epigenetic silencing, right? BRCA might be silenced by methylation. Okay? Right now, we can't detect it unless you specifically test for it. There are other genes, okay? Here. PARB2 we know about, RAD51C we know, but who knows what other genes are involved in the same pathway. But regardless of the specific mutation, they will all generate signature 3. Okay? And so, comprehensive identification of homologous recombination deficiency, which is related to PARP inhibitor, will increase the number of people who can benefit from this. Okay, okay, so what's the mathematical question here? The mathematical question is that when you have panel sequencing data, the math problem gets a little harder. So there was a paper from the Sanger Institute in the UK that used whole genome data to identify these signature 3 cases. There was a paper in Nature Medicine, and a lot of people figured out that that was really a good idea. But in reality, in practice, when you go to a hospital, they don't do whole genome sequencing, at least not yet. Okay? They'll do panel. So the question was, can we do that from panels? All right, so there are lots of samples that we could look. So there's a project called Genie, where multiple hospitals try to pull samples together. A company called Foundation Medicine has a panel called Foundation One. They are doing hundreds of samples. We are working right now with another company called Tempus that has done hundreds of thousands of samples. Okay? The problem is mathematically, when you have whole genome data, you have a lot of mutations, right? But when you have panel data, panel just means you only sequence a small number of cells, small number of genes, small number of genes that you're interested in. Okay? The data are much sparser. Okay? So the question is, can you look at, maybe 20 mutations and still figure out whether it's signature 3 or not? Okay? When I first saw this problem, I thought this can't be done, right? I mean, how can you figure out that this came from this rather than some other signature? Okay? Too hard, I thought. But though I figured out, the postdoc that I mentioned figured out how to do this. Okay? Now, our goal is right now, there is a testing you can do. So a lot of patients send out another sample for testing by this company called Myriad Genetics. It's $4,000. Okay, we thought, okay, well, we want to put that company out of business, or at least that assay out of business, because when you do panel sequencing, you can actually get it for free. Okay, everybody who does panel sequencing can get that information for free. I'm not going to tell you too much about the solution, except just a couple ideas. So one is that if I were to just look at that one profile, there's no way I can tell. But we know from a lot of genome sequencing data we already have, some correlation structure between signatures. Okay? They're not totally random. They tend to co-occur. Okay? Certain signatures like to occur together, and we can see this per different tumor type, because every tumor type is different. Okay? So we utilize patterns observed in previous data sets, and so this was not possible when we didn't have thousands of whole genome data. And then there's a little bit more of mathematical work that needs to be done. It turns out that right now, almost after 10 years, the software that most people use is based on the work of a PhD student at Sanger. Okay? And because they were ahead of everybody else, it just got adapted, but nobody could actually spend a lot of time until a few years ago to really think about whether it's the right mathematical approach. Anyway, so I will just mention the fact that the different tumor types are different in terms of their correlation structure. We take advantage of that, and then we use some fancy machine learning way to combine the data, and then we can actually do the prediction. And it turns out to be obviously not as good as whole genome data, but turns out to be pretty good for certain tumor types. Okay? And so what we find is that of all the cases that we think are signature 3 positive, which means HRD, okay, a lot of them are BRCA mutations, BRCA germline mutations. Okay? A lot of them have somatic mutations in BRCA, okay? But they are actually wild type for BRCA. Okay? They have mutations somewhere else, or BRCA genes are mutated by some other methods that's not a point mutation. Okay? And that in fact, if we look across samples, across tumor types, some of these cases have a lot of cases with HRD. Okay? And so for instance, you can imagine, I have a colleague doing pancreatic cancer research. You can imagine him saying, well, I know that some of my patients will benefit from PARP inhibitors, but which 5%? Right? You can imagine if you do a clinical trial where 5% of the cases benefit, it's going to fail, right? Unless you have tens of thousands of cases. Okay? And so that's why we're really interested in doing this. So we're working with many hospitals and companies, the hospitals and then potentially more companies. Pre-COVID, there was a lot of interest, and now it had died, and then now it's slowly going back up, to really make this a tool that everybody can use. And so we've actually done a lot of additional analysis to show that it works. I'll just, because of time, go through this rather briefly. We can, for instance, look at signature 3 cases that are BRCA negative, okay? BRCA positive, and then compare with the BRCA mutant cases, and they behave about the same, okay, etc. And we have data from clinical trials where signature 3 positive and negative groups have different survival. If you were to look at some other assays, so this is a functional assay that's done in the lab sometimes. This is the $4,000 test that you can do. Okay? We do better than all of them. Okay? So the commercial assay doesn't give you that separation. So we think that this idea of detecting signature 3 is a good one. And so once you have an idea, it turns out that even after you work out the method, it takes a lot of time and energy to actually apply it, because these clinical trial data are noisy. They are coming from multiple hospitals and different platforms. Just a lot of time spent on conference calls and authorship issues sometimes. A lot of, sometimes you have to spend your own money to do more sequencing. So there's just a lot of work to be done. And of course, when I first started doing genomics, perhaps you're in the same situation. You think of, okay, all these genomic technologies can be used to do some good, right? But that is actually much harder than you think. It actually takes a lot of energy that's not scientific. Okay? Just a lot of skills in working with people, a lot of patience, and a lot of paperwork, right, to get access to these data sets. Okay, let me tell you a couple other examples of signature analysis. So the latest of our methods development work related to this, I will describe in the second half, last part of the talk, if we have time. But I want to give you just a couple other examples. Predicting response to immunotherapy in mismatch deficient cases. Okay, I'm sure have heard of immune checkpoint blockades. Okay, it works really well for overall, across most tumor types, mismatch repair deficient cases. Okay, so that figuring out which ones are mismatch deficient, repair deficient, and proficient is not that hard. We can actually look at exome data and group them. Okay, so it was a big deal, right? That it works better for MMRD cases. But even then, okay, a large fraction of cases, depending on tumor types, 30 to 60% do not respond. Okay, so it's better than the MMRP cases, but not very good still. Okay, in fact, when we take a look at our data collected from lots of different studies, if you compare the mismatch repair deficient versus proficient cases, the survival difference is significant. Okay? But if you stratify within mismatch repair cases by all the things you can think of, they're not that helpful in distinguishing the groups. Okay? And so the question to us was, well, can we do better? Okay? Of these 55 that respond a little bit better, can we still distinguish the ones that do respond and do not respond? Okay. All right, so we do mutational signature analysis. Well, it turns out that couple signatures, 26 and 54, seem to be significant. Okay. 26, if you look at the catalog, it's one of the 7 signatures associated with defective mismatch repair, microsatellite instability. Makes sense. Okay. I'm going through this relatively quickly. But 26 and 54 all have a lot of T to C mutations. Okay? And it turns out when we do this with a set of about 55 cases, they do distinguish among the MMRD cases the better responders and the worst responders. Okay? And so we submitted the paper to Nature Medicine maybe 2 years ago. And then they all said, great idea, but you need more samples. Well, it turns out finding more samples, with genomic data of patients who have received immunotherapy is not trivial. Okay, there are a lot of papers. You go and figure out where the data are. You either cannot figure out what file belongs to whom, or they just don't have the data available. I mean, we've reached out to I don't know how many people. The most common thing I hear is, well, patients didn't sign the right consent forms. We can't share the data. Okay? Sometimes it's a pharmaceutical company that says, well, we'll make the data available, but we have to do the analysis. So you send us the code, we'll run it, and give you the result. Boy, that would be a long process, right? And so after about 2 years, we finally got some people to help us a little bit. And still, we could only collect about 40 more cases for validation cohort. Okay? Part of the problem in doing the analysis is that different trials measure slightly different endpoint. So this was overall survival, this was progression-free survival, and so on. So it's really hard to do the analysis. I mean, most of the methodology work was done 3 years ago, and now it's almost done. So all this is still unpublished. Hopefully it'll get submitted soon, and the reviewers will still be happy. I can tell you a little bit about what we learned from this by looking at the cases that have the high SPS 26 signature. We don't have expression data for all these clinical trial data, but what we do have is a lot of TCGA data, right? So we go back to TCGA, figure out which ones are SPS 26 or 54 high, divide them, and then do all this analysis that we typically know how to do. So pathway analysis shows that DNA double-strand break repair is an active process that's related to this. Okay? I won't tell you all the details. If you are interested, you can ask me later. But we can actually come up with some explanation based on our mutational signatures why this is happening. Okay, just one other application, and then maybe I'll talk about the methods part really briefly. So another thing that we thought of to do with our single cell techniques is to look for off-target mutations from CRISPR editing. Okay? I'm sure you've heard of how CRISPR editing will transform certain therapeutic areas. I would say to those company representatives, how do you know that there are no off-target mutations? Well, what they typically do is look for places that they think matches the intended sequence, right? So you have guide RNAs, and so if you assume where the off-target mutations might occur and sequence those, okay, many papers have done that. And, maybe you can claim that there are no off-target mutations. But in fact, people have not done genome-wide mutation analysis. Why not? Because if you have a population of cells and the mutation occurs, the off-target mutations are not going to occur at the same position, right? Unless you sequence, so deep that you capture every cell, you can't find them. And so there's a major company that claims that there's an off-target mutation. They've done 500x whole genome sequencing of the bulk. Of course, they're not going to find anything. And so I've always been a little skeptical of the idea, but we didn't really know how to test, how to look for these mutations, okay, until recently. So what we did was, now that we know how to amplify single cells and look for point mutations pretty accurately, that took many years. There are other applications including this. And so what we've done here is work with George Church, who's in the genetics department at Harvard, to do some base editing using a system that they've developed to do multiplex editing. So multiplex editing means you're actually trying to modify multiple sites. Again, the topic is mutational signatures, so I'll just mention that part of it. This could be a whole other talk. But briefly, we've done some experiments to figure out what the off-targets are. So as most of you know, editing efficiencies are not perfect, 100%. Okay, we know this. And then, as I said, it's a challenge. Identifying these off-target mutations is a challenge. Recently, there was a paper from David Pellman's group. He's a colleague of mine at Dana-Farber. He actually showed by doing single-cell DNA sequencing at very low coverage that there are all these structural alterations. Okay? And so because other people hadn't done it at single cells, they just didn't find it. Okay? And so the question was, can we examine mutations at the single-cell level? Okay? And this was mostly Vinay, who's a graduate student in the lab. Okay, well, because of time, I'm not going to tell you all the details of how we do this analysis, except to say that compared to wild type, okay, we see when we have these editors added, you see more mutations, and this group is on-target mutations. But when we look at places away from the intended target site, we still see quite a bit of mutations. Okay? In fact, we see on the order of 150 mutations per cell. Okay? Now, is that going to be a major problem? Probably not. The genome is a big place, right? It probably won't make a huge difference unless it hits some important, regulatory region or some exonic region, right? And so we don't really know exactly what the functional consequences are, but we should be aware that such things can occur. So after we found these mutations, we can actually do some analysis of where they occur and in what context they occur. And we find that SBS 8 and 5 are prominent, and SBS 8, if you look at the catalog, it'll say it's unknown origin, but it's actually related to nucleotide excision repair by many recent papers. And we have some other data to suggest that it's similar to what we see in germline cases where that pathway is mutated. Okay? And so, again, by using signature analysis, we can actually infer the mechanism, right? Otherwise, it's just a bunch of mutations, right? Okay, so the lesson then is just that whether it's somatic mutations in cancer or mutations in any other type, you can try to do this mutation signature analysis. And the fact that you have, in many cases, very small number of mutations means that you need to have a methodology that can handle such cases. Okay? If you have, like in cancer, if you have, 10,000 mutations, everything becomes easy. Okay? If you're listening to that music for an hour, yeah, it gets pretty easy. But the shorter the duration is, harder it becomes. Okay, I'm going to just tell you in about 5 minutes just one issue with the standard analysis, and I won't talk about how we resolve that, but let me just tell you what the issues are. Okay. Okay. This is something that we had a summer undergraduate do. It's pretty simple. Okay, so if you think, the idea of mutational signature analysis is that you have multiple processes that generate the mutations, you combine them, and you try to infer the original processes. Can we just try some simulations? So SBS3, and then some other ones. So this is the harder one because it's kind of flat. It's harder to distinguish. So the simulation was we mix them at some random fraction, we simulate data, and then we try to see whether we can actually recover these. Okay? Well, it turns out that every time you simulate and try to recover, okay, you get slightly different results. Those are the dots here. Well, it turns out that every time I do this, I can recover signature 12, 8, and 23. These are kind of spiky. But every time I do it, I don't quite get signature 3. I get something slightly different. Okay? And so cosine similarity, you might remember from your linear algebra class. If I see, if the algorithm gives you these 4 things, I can say, well, actually this one matches 23. Okay, so I found that one. This one matches 12. Okay, but this one doesn't quite match SPS3. In fact, if I try to force it, okay, if I look at the catalog and try to find some combinations that matches what the algorithm gives me, it gives me signature 3, 23, and something else. Okay, because mathematically, I'm trying to figure out from this what the combination of the original signatures is. Okay, and so sometimes these things just sneak in. Okay, just because the data are not perfect. Okay? Anyway, so we have some new algorithm that can fix this sort of problem. Okay? Okay, I'll just end here and take questions. So the point again is that mutational signatures is pretty useful, and that in fact, if you identify an interesting problem like the one we did for panel data, it gives us a chance to think of a new way to reframe the question so that we can actually do something useful. And again, it's like any other method. False positive, false negative signatures can arise, and so sometimes we will look at a paper and argue whether it's an artifact or not. You can imagine that sometimes people will want to give a therapy based on some signature that they think they find. Okay, so you really want to be sure that it's there. Okay, that you're not guessing. Okay, just to actually tell you, I have lots of funny stories, but tell you one funny story. Some months ago, I was called to be a juror in a jury trial. So in the U.S., we have a trial where a bunch of people, common citizens come and do this case. I was called, and I thought, oh no, I have to, be away from work. the trial was about a smoking case. It was an old woman who died. Her family was suing Philip Morris, okay, the tobacco company, and they said the trial was going to be about 3 months, okay, which means every day I have to go to the court for, 9 to 5. And I thought, oh my goodness, my wife is going to be very, very unhappy. And what about, people in my lab? And then the way it works is that anybody who has a reason to get out of the trial can, raise hand, and the judge will one-on-one describe. He knew that a lot of people would be disqualified because, for instance, if your mom had died of lung cancer, you might have a bias against the company. Anyway, so I said, okay, for someone to prove that, for the petitioner to prove that the death was caused by smoking, you need to signature analysis to figure out what the source of the mutagen was. Now, unless you do that, in fact, of the lung cancer cases, about a quarter of them are from non-smokers. Okay, so how are they going to prove really? Because they're asking for millions of dollars. How are they going to prove that this woman died of smoking? Okay, and in fact, there have been other cases where you can imagine someone suing a factory saying, oh, the factory caused my cancer. What's the mutagen in the company? You should sequence the sample, the right tissue, and do signature analysis. Anyway, so I was lucky, and the judge said, okay, you're out. Anyway, so again, it's a delight to be here. I'm happy to take questions, and after that, I can stick around and answer more questions. All right.